So in order to obtain an experimental measurement of KD, it's necessary to measure concentrations of the complex versus the function, or as a function of L. This is usually done in two ways. One, monitor a change in property of the protein. Could be of the protein, it could be of the ligand itself, could be of them complexing. Okay. And then the other one is something called equilibrium dialysis, which your book doesn't talk about at all, but I wrote a homework problem about it, so we'll talk about it briefly. Okay, but basically what we're gonna have is we're gonna have a beaker here. It has some, probably just water as our solution in it. It has a dialysis tubing bag. Inside are our proteins. Okay. They can't get out, okay? The pore size in the membrane makes it so that the proteins are too big to get out. So this is our concentration of protein. I'm going to add to the outside my known concentration of ligand, which is small enough to get through. And I'm going to wait for equilibrium. And so now I'm going to have a situation where my proteins are bound to their ligand. Also, the concentration of free ligand inside is going to match the concentration of free ligand outside, okay? Whatever's unbound in and out is going to want to be the same given enough time. Okay, we already talked about chemical potential difference or concentration differences. Okay, so there's both the bound up stuff and the free stuff. Inside our dialysis tubing then, we have the concentration of the complex, plus we have some free ligand. We can take a sample out. So we put in some known concentration of ligand here. After some amount of time, that concentration in the outside has gone down because some flowed into the dialysis tubing, bound to the protein, and then also what was free in solution wanted to be equal on the inside and outside. So if I take a sample on the outside, I have some concentration of what is now my free ligand. I know what I started with. I know what this is. It's going to be the same as the outside. So I can calculate how much bound up, and I can solve for this piece. Okay. So that's like a very simplistic, this is just in words an explanation of that. It's a really easy experiment to run. And again, you could do this, you can change the initial concentration of ligand over and over along that x-axis so that you can look at how this concentration changes and get yourself a dissociation constant. And so the homework problem is easy. It just puts numbers to all of these things. You've put in 11 micromolar to the outside of your dialysis tubing. You wait this amount of time. Now it's down to 5. What has happened? I think you guys will be fine. Okay. And this article here just gives a nice overview of a whole bunch of different types of binding assays. So I think it's good reading, and I know last year I definitively pulled at least 1 or 2 figures out of it and put them on the exam, just to have you know, it'll be fluorescence or something on your y-axis for your protein complex. You'll have varying ligand concentration, and I'll ask you, read the KD off of this. You know, which of these 2 ligands in this figure was stronger? How do you know? So make sure you know how to do that stuff. Right. So let's look at a specific example from your textbook now. It's estrogen binding to its receptor. Okay. So estrogen is the ligand. It binds to, it is a type of steroid hormone. So it's going to bind to its receptor, which happens to be in a family of proteins that are specific for binding to DNA. Okay. The super simplified version of this is there's the receptor, there's estrogen. When they bind, once estrogen binds to its receptor, it will then bind to DNA and initiate the synthesis of other proteins. The most oversimplified version there can be. A slightly less, but still very simplified version of this situation is normally what we'll have, so here's the domain that it can actually bind to DNA. All right, here it just looks like it's a 1-domain receptor, okay, but it's actually a complex of many things. So there's the piece that can actually bind to the DNA. There's the piece that's actually going to initiate transcription and activate it. There is a piece that's responsible just for hinging and actually changing the conformation upon the binding of estrogen. There's the site meant for estrogen to bind. And there's also an inhibitory piece. So estrogen comes in, when it binds, this inhibitory piece has to dissociate. The inhibitory piece is there to prevent this receptor from binding to DNA and initiating transcription when we don't want it to, okay? It's in the way. So estrogen will bind, the hinge region will undergo the conformational change. It moves such that the inhibitory piece can get out. Now the DNA binding site is exposed. It'll come down and bind to DNA. The actual transcription domain will initiate transcription. And I say this is only slightly less simplified because this is showing maybe 5 parts. The actual complex has over 20 elements interacting together. So I really don't dream of being the type of people that learned all this stuff in the first place. That's just, but it's very, very sophisticated. Okay, so the receptor undergoes a conformational change that exposes the DNA binding site and can regulate downstream transcription. So let's talk about how we would do a binding assay to get the case of D for the estrogen binding. We're going to start with estrogen, and we're going to add a radioactive label to it. Okay, this is kind of hard to see. This says tritiated estrogen, or it's shown as H's with a little asterisk over them. Okay. So tritium is a hydrogen isotope. 3H, it's 1 proton and 2 neutrons. Okay. It's about, I don't know, it has like 3 times the mass of regular hydrogen. So that's the radioactive isotope we're going to use, and we're going to tag all of the hydrogens on our estrogen with this isotope. Then we're going to mix our radioactively labeled estrogen with our receptor. We're going to let the system come to equilibrium. So we're going to wait for the binding to occur, and then we're going to separate. We want to get apart the free ligand that didn't bind and what is bound up to its receptor. Okay, and so here's kind of an illustration of that. We have a solution of our receptor. We have a solution of our radioactive ligand, our radioactive estrogen. We mix them together. We wait some amount of time. Now, in this particular case, there are several ways to get them apart, these separating parts. There's a lot of different ways to do this. So this is the first example. We'll show another example, and later I'll show a third way. Okay, and just what you need to use to separate your free and bound ligands, what experimental technique you choose, will depend on what you're working with. So in this case, you'll add some negatively charged resin in here to bind up the receptor, and then you can use centrifugation to pellet all of the receptor that's bound and remove all of this free unbound estrogen that you're not interested in, okay? And then you could save your supernatant. All of this has what was the free estrogen. You can measure what's there. And you're going to get a pellet of all the bound up estrogen bound to the receptors. Now, why does a negatively charged resin work in this case as my separation technique? What did I just say that the receptor wants to bind to? Eliana.
The receptor can be positively charged, so it hasn't binded.
It may have positive charge on it that helps it have this interaction, but what did it want to bind to once estrogen was bound to it naturally? What was the normal cascade of events that happened? It bound to DNA, and DNA is negatively charged. So if I can put something, and it possibly has positive charges that influence that. There's probably many contributing parts of that binding site. But since DNA is negative and I know it would want to bind DNA, that's what it's going to go do. If I put something else negative in there, the receptor, once it's bound, will bind up to it. But free receptor won't bind to it because it doesn't have its estrogen bound and it doesn't have its DNA binding site available. Okay. So that's why we'll put in the negative, spin it down. I now have both my leftover stuff that didn't bind and a pellet of all my attached stuff. Okay. So then you can measure the radioactivity of this sample here in the pellet to get a measure of how much estrogen bound to the receptor. The other option would be to use a filter to track the protein and the protein complex, some filter that is small enough to let free ligand through. but too small to let proteins or the protein complex through. So just as another separation technique, right? Let all of this go through, and then now what you have is just your protein bound to the estrogen. Okay. So measure your radioactivity in your pelleted sample with a liquid scintillation counter, which I don't really know how that works, other than you're going to have some incident radiation that you hit it with, and you're going to measure the resulting light pulses, and all the interior of that instrument is for someone else to teach you about, or you can look it up on your own. So radioactivity will be proportional to the concentration of the protein ligand complex, right? We put radioactively spiked estrogen in, some amount of it bound to the protein. We threw away all the leftover free estrogen, so that's not there contributing to the radioactive signal. The protein's not radioactive by itself. So any radioactive signal we measure is exclusively from estrogen that managed to bind to a protein. And then I say lather, rinse, repeat, because we're going to do this at a whole bunch of different concentrations. We're going to go from like 1 nanomolar to 80, and we're going to repeat this over and over and over so we can build our binding curve. I don't know if anybody works in a lab. This type of stuff is so tedious and takes so much time. It's not the most fun, in my opinion. So here we go. I want to make my binding curve for estrogen to its receptor. I'm going to repeat this assay for increasing concentrations of ligand until I reach a plateau, until I hit my saturation point, and then I know I've bound up all the protein that there was. Okay. So down here we have concentration of free ligand in nanomolar, and we have our concentration of our estrogen bound to our receptor. This happens to be in picomolar. These will depend on what species you're working with, what kind of order of magnitude you're at. And you can generate your curve, and you can find the halfway saturation point. Once you have saturation, you can find your midpoint and calculate this, which is about 5 nanomolar for estrogen to its receptor. So it's a pretty strong affinity. Like we said, nanomolar is the range of high affinity.